All right, so next up we have Dr. Randall Roper. Dr. Roper is an associate professor of biology and an adjunct associate professor of biomechanical engineering at IUPUI. He has a BS in microbiology from Brigham Young University and a Ph.D. in immunology and genetics from the University of Illinois. His postdoctoral fellowship is in genetics and development, and it was completed at Johns Hopkins University School of Medicine. Dr. Roper is currently involved in research that seeks to understand the genetic and developmental basis of phenotypes related to trisomy 21. His research hopes to better define how the 3 copies of the 21st chromosome affect traits associated with DS, as well as the severity of those traits. So welcome, Dr. Roper.
Thank you, Stephanie. And Stephanie Sherman mentioned that it was hard to follow Carl Erskine, and I'm going to mention that it's hard for me to follow Stephanie Sherman, who is a fantastic researcher and has really done a lot for the Down syndrome community. And I just want you to know that the speakers and the individuals who are not speaking are some of the world experts in Down syndrome, and we are very lucky to have them here. And I'm grateful to be here and to be a part of this conference. So some of the goals of my presentation, really to give insight into how a scientist thinks about Down syndrome. And I will do this by examining one of the current potential treatments that is being talked about in the Down syndrome world, and that is green tea, the green tea polyphenol, and some of you might have heard of this, EGCG. I will also review some of the other treatments that are used, and I really hope to leave with you, and I hope to paint that there is really a bright future for individuals with Down syndrome and for understanding why individuals with an extra chromosome 21 really have a bright future to look forward to. So I wanted to start by saying this might be what the parent's perspective of an individual with Down syndrome. You see your child, and many of you have done a lot, and all of you have done a lot to help your children, to make sure they're getting the best care possible, the best education possible, and you are really thinking about your children and their best, and have their best interest in mind. Now, when you think of when a scientist thinks about Down syndrome, this is what I think about. Those just happen to be the list of genes that are found on human chromosome 21. So we think a lot about why do 3 copies of these genes, how do they affect people with Down syndrome? Now, I have a couple of disclaimers that I want to give before you think before you characterize me as who I am not, okay? First of all, I am not an MD. So any individual that I treat has 4 legs and walks around, and most of you don't like these creatures, but I treat mice. The second disclaimer I have is I do have a niece with Down syndrome, and when I get really tired of the laboratory, I take a break and go to Chicago, where I can refocus my priorities. And Megan is 22 and is entering an exciting time in her life. So I get to see kind of the world from both sides. And what I think about is, as Dr. Sherman mentioned, how 3 of these copies of genes on human chromosome 21 affect traits that are seen in individuals with Down syndrome. So when you read something like this, or when you come across something like this on the internet that says a pill for Down syndrome, new research indicates that a treatment may one day lessen the symptoms of the disability, you think about how this might affect your child, or friends of your child, or individuals with Down syndrome. And you say, wow, what are the implications? What a scientist thinks about is, where's the scientific literature? Okay, where is the research behind this claim? And so the first thing that I do when I see this is I go to a place that's called PubMed, and I say, Where's the literature on this? So I'd like to, in our conversation today, I'd like to talk a little bit about the scientific process and really where this all fits in. Well, first of all, someone might have an idea, and this can come from a scientist, this can come from you, this can come from different areas of research. You know, when we have some anecdotal evidence that something might work, it's usually some positive evidence. We don't talk a lot about the negatives, and that's actually a little bit of a problem. And usually this idea that we have is based on something that we understand, a mechanism or something that we can understand as scientists. So then scientists, such as myself, we will go to the laboratory and we will try to understand a mechanism. We will propose a hypothesis. If this happens, then we expect this to happen. And as Dr. Sherman mentioned, we use a lot of mouse models. And so that's really where we can understand a little bit more about where these ideas could take us and the possibilities. And then these ideas might progress to a clinical trial. And there are different phases of clinical trials. We have phase 1, just looking at adverse effects and seeing if there might be a possibility to have an effective treatment. Phase 2, we use them on a small group of individuals. And phase 3 is used on a larger group of individuals. And then finally, we might have a treatment. And you'll notice that I've kind of put the people who are involved in these different processes. And I think it's very important that we understand that these processes take a lot of time and take a lot of manpower. And the individuals who will be speaking today are involved in different levels of these processes, and they're all very important. And the one part that I don't want to leave out, and I make sure that I don't leave out, is you. That you as parents, you as caregivers, you as people who know individuals with Down syndrome are part of that process, really in every step of the way. So, when you find something on the internet, and this has been seen, this is from the Huffington Post, as well as CNN, green tea compound can improve brain function for people with Down syndrome, study finds. And another one, can green tea boost your brain power and treat disease? And then it talks about how compounds in green tea may be able to help treat symptoms of dementia and with individuals with Down syndrome. So you read this and you say, wow, is there something in this green tea? Or what is everybody talking about? So when I read something like this, and it might come to me in a situation, in fact, I did read something like this back in 2008. And it was a study that was done by some scientists in mice that said green tea polyphenols rescue brain deficits associated with Down syndrome. But what I was really interested in is that they targeted a gene. So one of the genes in 3 copies found in individuals with Down syndrome is this gene called DYRK1A. So we talked about this, and we thought about this in the lab for a long time, and we talked about it, and we said, is this something we want to try? And is this something, do we think there is merit in looking at this particular polyphenol that's included in green tea, a polyphenol called epigallocatechin gallate, or just EGCG for short. I promised the translators that that would be the only time that I would use epigallocatechin gallate. So we talked about this, and we decided there was some merit because there were scientific studies that said that EGCG did indeed, in vitro, lessen the effects of what this gene DYRK1A was doing. And so we went back to the laboratory. And what did we do? Number one, there was an idea. This idea was brought out in the research. It was brought out in the popular literature. So we had the idea, and we said, we're studying skeletal deficits. Would lessening the effects of DYRK1A help improve skeletal deficits? Now, as Dr. Sherman mentioned, that there is increasing longevity in individuals with Down syndrome. Individuals with Down syndrome have a shortened period of adolescent growth. They have generally short stature. They have reduced bone mineral density, lower rate of bone turnover, increased fragility, and as they age, they seem to have an increasing incidence of osteoporosis. So as basic scientists, we used this mouse model of Down syndrome that was already explained, called the TS65DN, that has about half of the genes that are found on chromosome 21 in 3 copies. Now, I want to talk a little bit about Down syndrome, and Dr. Sherman gave about mouse models of Down syndrome. Dr. Sherman gave a great introduction to that. There are, as she mentioned, a number of mouse models of Down syndrome, and this is one way the research area is progressing. And so we have different sets of genes that are found on chromosome 21 that are found in different mouse models. But just as important, we have traits that are found. So traits that are associated with Down syndrome that are found in mouse models. And so we have behavioral traits, craniofacial traits, developmental, heart, things related to cardiac defects, learning and memory, and so forth. And we hope from these mouse models that because there is a similar construct, so because there is a similar construct in the development as well as the genetic component of our mouse models, as well as these mouse models tend to imitate the traits that we see, so they are good models of the traits involved in Down syndrome. We believe these mouse models will also have predictive value, will help us understand a little bit more about Down syndrome. And so in a paper that we published a number of years ago, we established that in the TS65DN Down syndrome mouse model, that there were skeletal deficits, just as you see in individuals with Down syndrome. And if you look very closely at the picture on the left, well, actually, this is a femur. So basically, this is your leg bone. And if you look at the bottom where I have the red slash, so this is what we call the distal end of the femur on the right-hand side. And what you have is you see developing bone that we call trabecular bone. And this is what will develop the bone and help to give it strength later on. And as you look in the diagrams, and as you see in the diagrams, there is both a normal mouse as well as a trisomic mouse, and you can see that the bone on the left-hand side is not as prevalent in the Down syndrome mouse or in the trisomic mouse at both 6 weeks and 16 weeks of age. So there's a problem with how this bone is developing. We also showed, and as you look at the cortical bone, and that would be the bone that's basically in the center of the femur that gives the bone its strength, that there is less cortical bone. And therefore, the bones in this mouse model of Down syndrome are less strong, and they tend to break easier. So we established in our mouse model that there were these bone phenotypes that accurately represented what is found in individuals with Down syndrome. So we wanted to understand the role of one gene, this role of DYRK1A, and how it affects these skeletal phenotypes. And so we thought, well, if we could lessen the effects of DYRK1A with a known inhibitor, we might be able to correct some of these bone deficits. And the next thing that we can do in mouse models that we're not able to do in humans is, what if we take out one of the 3 copies of this DYRK1A gene in our Down syndrome mouse model and reduce it to 2 copies? What will it do to the skeletal effects? And so we hypothesized that we could use EGCG, this green tea polyphenol, to correct bone deficits in the skeleton in our mouse model of Down syndrome. This is what EGCG looks like. It's the most common green tea polyphenol. It's a potent antioxidant and radical scavenger. It crosses the blood-brain as well as placental barriers. It's been shown it's been used in treatments for Alzheimer's disease as well as Parkinson's disease. And most importantly for us, it is an inhibitor and has been shown in vitro to be an inhibitor of DYRK1A, this gene in 3 copies. So this is what I like to call what I did on my Christmas vacation. This next part is I decided I'm going to try this. And so we fed 3-week-old mice EGCG. We gave them about 10 milligrams per kilogram per day, and then at 6 weeks we analyzed their bone. I had a graduate student that was working for me, and I tried to get him to do this project, and he said, that's just going to be a waste of time. But I said, Josh, we've just got to try it. We've got to see what it does. It's either going to work or it's not. And so I did this over my Christmas break, and then I talked to him afterward, and I said, I think you'll want to see this because the mice are actually bigger. And he kind of looked at me, and all of a sudden this became his Ph.D. dissertation project. Because what he found, if you look, once again, we're looking at trabecular bone. This is the bone that is forming. You see normal mice, and in the second picture, you'll see our Down syndrome mouse model. And in mice that we gave 3 weeks of EGCG, you see more trabecular bone. And the graphs below are showing that. And so we say, oh, this concentration of EGCG might help improve bone in individuals with Down syndrome. But what it didn't do was it didn't improve the cortical bone, that part of the bone that gives the bone strength. And so we said, we might have something here, but it's not yet good enough. And we also looked, and it didn't improve the strength of the bone. So the bone, even after the mice were given EGCG, broke just as easily. And so we said, we've got some work to do. And so we said, let's see what happens when we reduce the DYRK1A genetic copy number from 3 to 2. And so this was the genetic hypothesis. We hypothesized that if we reduced it from 3 to 2, all during the lifetime of the mouse, that it would improve the skeletal deficits. So how do we do this? We can breed mice that only have 1 copy of DYRK1A with our Down syndrome mouse model, and basically down below, as if you look in the second picture, there is a mouse, a trisomic mouse that only has 2 copies of DYRK1A. And we examined this mouse, and we found out in the hashed mark there that it had increased bone mineral density at 6 weeks of age, and that it had increased trabecular bone at 6 weeks of age, and that it had increased bone strength. So like scientists, like good scientists, we published this. And when we say we published this, we sent it out for peer review and asked great scientists all over the world to look at this and see if we're on the right track. And if there wasn't something that was right, they would ask us about that. And so we published this, and we established that DYRK1A was an important contributor to skeletal deficits. Now, when we published this, we wanted to get this information out there, but we had a worry. And the worry was that people would skip some steps and start giving their children EGCG to increase their bone strength, or to say, I'm already giving my child EGCG. It's going to help them, right? Well, EGCG is widely available. You can pick it up on the internet. You can pick it up at natural health food stores. And so we said, wow, we're going to publish this, and we have a worry, and we have a hope. And our worry is that everyone is going to start giving their child EGCG and will believe that it improves their bone. And now, if you look at the study very carefully, it improved 1 parameter of bone, but not really the strength. But we had a hope when we said, but we might be able to give EGCG for a little bit longer, or perhaps a little bit higher dose, and maybe it will actually improve bone strength. And that was our hope. So what did we do? We went back to the scientific process. We went back to basic science. We went back to the lab. And we went to blogs of people that you might read, or you might have a blog, and we said, what are people saying about EGCG? And we found our material on blogs, and people were talking about us, and we said, we better do this quickly. And so what we did is we looked for the supplements that were containing EGCG that were mentioned in all the different blogs, and we went out and we found them on the internet and we ordered them. And then we started to compare them. And I'm not giving away any names, but if you're using a supplement with EGCG, it's probably up here. It's just coded, okay? And we said, how much EGCG is there really in there? And and what else is in there. And so we looked at this. Now, something that you buy at a health food store is not regulated by the FDA. And so one of the first things that we looked at is what's in there, actually what it says is in there. So what we did is we looked at pure EGCG, and we have ways we can purchase pure EGCG, and we can quantify it and make sure it is pure. And in fact, we have purchased EGCG that was not to the level that we expected it to be, and we sent it back and we said, it's not good enough for us because we want very pure EGCG. And then we compared it with two different supplements, one that had 50% EGCG with 98% total polyphenols, 80% catechins. Okay, so it had all these EGCG plus these other things, and then one that said it was 90% EGCG. And what do? We gave them to our mouse. Same thing that we did before. So we gave these supplements along with pure EGCG, different mice. The mouse would either get pure EGCG or one of these supplements or just water, a placebo. And we looked at their skeleton at 6 weeks. The first thing that we found is when we characterized the EGCG in supplements, we found that a lot of times it wasn't what was advertised. There wasn't the amount of EGCG in the pill that people claimed it was. And when we put it in water, it tended to disintegrate very quickly or change into something else. And that concerned us. Well, we gave it to our mice, and we looked at the trabecular bone. Remember the bone that was forming? And these two supplements improved the trabecular bone. And we said, wow, that's pretty amazing. But then we looked at the mechanical properties. We broke the bones. And the mice that had received these two supplements that contained EGCG were worse than our Down syndrome mouse that had just gotten water. That was shocking. But we don't know what else are in these capsules. We don't know what else is going on. So we published this. This went through peer review, and we established that all these supplements that contain EGCG are not created equal, and they may actually harm people that are taking them and harm their bone. Well, different people have proposed that reducing the effects of DYRK1A will help with cognitive impairment in individuals with Down syndrome. In fact, there have been studies that have shown that in mouse models that it improves some of the cognitive deficits. And some of you may be familiar, these supplements with EGCG were given to same Down syndrome mouse model that we used. They were higher than we used for our skeletal treatments, and there were certain tests that they seemed to improve the mouse's cognitive ability when they were given these supplements. Two of these tests were the novel object recognition test and the Morris-Water maze, and we don't have time to go through that, but if you'd like to know more, I'd be happy to explain about these tests. And further, these scientists started to do clinical trials with EGCG. And some of you may have heard that in a phase 1 type clinical trial, these individuals, these scientists gave a supplement. Now, it was very interesting to us what's in that supplement. So it's called mega green tea extract, lightly caffeinated, so it has caffeine in it. It's 98% polyphenols. It's 45% EGCG. So we don't know what's in the rest of the 55% or 50% besides other polyphenols. They gave it to individuals with Down syndrome from the ages of 14 to 29, and there were some effects in some visual memory recognition tests that they did. And as you can see, these lines kind of at 6 months, they kind of separate, but you can also see that the lines we call error bars are actually overlapping. And so those effects were very small. There has been a second clinical trial, more of a phase 2 clinical trial, that has been published with EGCG, and it shows improved cognition. In this clinical trial, EGCG, this mega green tea extract, this time without caffeine, was given to individuals with Down syndrome, and they included cognitive training. Now, they had some in every clinical trial that you do, you have some primary outcomes that you're looking for. None of their primary outcomes were met, so they didn't improve in the areas they thought they were going to, but they improved in some secondary outcomes. Here you can see in some parts of inhibitory control and recognition memory and some behavior. There were some slight improvements after, during the treatment as well as after. So we said, but they aren't actually giving these individuals EGCG, just straight EGCG. It's EGCG with something else. And we said, so what happens if we go back as basic scientists and give the same amount of EGCG, just pure EGCG, that they did in their original studies to our mice? And so we gave an equal dosage of pure EGCG in our mouse model. We treated for a longer period of time in older mice, and we saw very minimal improvements. So we did not see the improvements that they saw, number one, in their mouse model, and we kind of saw the same thing, that there were very tiny, very small improvements. Well, the other thing that we saw is that this higher dose of EGCG, of pure EGCG, and we're talking about 50 milligrams per kilogram per day, actually weakened the bone structure. And based on all of our tests, it hasn't had any effect on DYRK1A. And so if there is an effect of EGCG, it's doing something else besides involving DYRK1A. So our conclusion is that EGCG may not be the best thing to be giving individuals with Down syndrome. And there may be, in some of these other treatments, in the mega green tea extract, there may be other molecules that might be helpful, but we don't understand what those are. And so one of my messages is to you is, if you think like a scientist, you have to be careful. You have to be a little bit careful of what you're doing and really read the literature and understand the literature. And I compliment all of you for being here today, because you're going to hear what's on the forefront of science. And hopefully this will affect how you think about this and what you think about with individuals with Down syndrome. So what about this treatment process? Where does it come into play? You know, you and I can go look on the internet and you can find a lot of claims for things that this will help and this will help, okay? And what I would say to you is, the first thing I say is, look at the scientific literature, okay? Remember, everything on the internet is not true. Do what you're doing today. Be well informed, attend conferences. Read the fine print. What is the EGCG that they're actually giving? Ask smart questions and ask your doctor. So talk to your doctor when you have a yearly checkup or whatever. Now, what I am not saying is, I'm not saying to not participate in trials, because one of the best things that we can do, one of the best things that you can do, is get involved in the clinical research. Get involved in the bona fide clinical research that you're going to hear about today. Not just this research, but other scientists who are really working on these problems. And I think what we really have is a bright future to try to find potential ways to improve the lives of individuals with Down syndrome. I've seen in the past 15 years a renaissance in individuals studying ways to improve the lives of individuals with Down syndrome. And so here are a selection of the drugs that have been tested for mostly cognitive phenotypes in individuals with, these were tested mostly in mouse models, and some of them have progressed to cognitive phenotypes. So there have been over 20 of them that have successfully rescued some type of brain deficit in our mouse models. Some of them have progressed onto clinical trials. Some of them are even targeting genes on chromosome 21. And this is really where I feel the best way to go is to actually target a gene product that is overexpressed. And that's what we're concentrating on in our laboratory. there are some caveats. Not everything that works in a mouse works in a human. In fact, this happens for all types of drugs. So it is known that there's a high failure rate of clinical trials that are based on mouse models in general for any disorder. But yet we can understand a lot from a mouse. And as most of you are aware, there was a phase 2 clinical trial conducted by Roche that was stopped. And because it wasn't achieving its aims. But despite that, we have a number of very intelligent and very savvy researchers working on understanding deficits, understanding traits that are found in individuals with Down syndrome, and Dr. Sherman explained that very well. We have pharmaceutical companies, and I think this is very exciting, that are becoming involved, and we are learning from the things that we have done and others have done on how to improve and how to test different molecules. And as has been mentioned before, Vince mentioned this, there is really a national, international emphasis on increasing research in individuals with Down syndrome. And so I encourage you to think about the scientific process. It does take some time. It starts with ideas. There's a lot of basic research. There's a lot of clinical research that goes on before we can come to a treatment. And I think what is really important is that we all realize our roles in helping to understand Down syndrome. I, as a scientist, have a role in the basic science part of this. Clinicians have a role, and we have clinicians here today that are looking and testing and trying to understand Down syndrome. You, as caregivers and as individuals involved in the Down syndrome community, have a role. And so I encourage you to become involved. If there are trials that you can become involved in, become involved and try to do your best, because this is the way together that we'll be able to understand more about Down syndrome and hopefully paint a wonderful future, not only for the children and the individuals that you know, but for those who are coming along in the future. And we hope that this will be a bright future for everyone because of the research that is being done in Down syndrome. So I thank you for your attention. These are some of the individuals that have been involved in some of the research that I presented today. And I will be available all day to answer questions or to talk more about this. And so I would be happy to take questions then. And thank you very much.